Okay, good morning, everybody. Thank you for joining us. We have a couple important topics, very important topics to speak about today, and of course, our main presentation on vaccinations. As usual, we're going to just give it about a minute or so for all of you to populate the attendee list here. And of course, as usual, we will be sending out a CME link. I'll send that out in just a couple of minutes. And so thanks for joining us. Upcoming, we have a lot of great grand rounds in the next few weeks coming up. And one of the ones we've been working on for quite some time, which is a very important topic, of course, is the health equity in Hispanic and African-American communities. That's going to be led by Dr. Fernando Mendoza and Dr. Kenji Taylor, both at Stanford. And we're really excited for that topic. The week after next, we'll have a combination of Santa Clara County response and East San Jose experience. We have Dr. Elsa Villarino. She's the Assistant Health Officer and Tuberculosis Controller at the Santa Clara County Public Health, along with Dr. Alex Studmeister. He's the infectious disease doctor over at San Jose Regional Medical Center in East San Jose, perhaps the busiest hospital with COVID-19 infections in our county. So they're going to be sharing experiences, and we're going to get a chance to learn a lot from them. Okay, great. People stand. I'm going to turn it over now. Dr. Harrington, our chair of medicine, would like to say a few words.
Yeah, thank you, Errol. And before I make a few remarks, I just want to thank our guests for joining us today. It's really been terrific to have colleagues from across the campus over the last few months join us in Medical Grand Rounds. So welcome and thank you. And I wanted to take a moment today to just reflect in the midst of Medical Grand Rounds, which we consider to be a special tradition in the Department of Medicine. It really gives us an opportunity to come together weekly as a community. We share ideas, we learn, we discuss, we argue, we debate with each other. But it's a very important event in an academic community and in an academic Department of Medicine. But before we launch today in talking about science and clinical medicine, I thought it was important that we pause and really think about what's been going on these past 8 days in our country. And I personally, as a physician, as one of the leaders of this department, really felt it important that we take this moment and pause and think. As many of you know, I'm a cardiologist, and so this might have special meaning when I say this. My heart hurts this week. And the reactions to the brutal murder of George Floyd are the expressions of people across the country who've been pushed past the breaking point. And it's been horrific to watch. It's been sad. But I think it's been important for us to really take pause and to think about why this is happening. In the midst of all of this, we're in an infectious disease pandemic. And the COVID-19 pandemic has certainly laid bare the health inequities that we have in our society. There's never been a time that the social determinants of health have been exposed so clearly, so rawly. People in vulnerable communities are dying from a disease because of their race and because they're poor. That should not be acceptable in the richest country on earth. I've asked, and I've been asked a lot, including by people that you see every week on this screen, what can we do as a Department of Medicine at Stanford University? I honestly don't know the answer yet, but I can promise you this. We can't go back to business as usual. That's not appropriate. It won't be who we are. Some of you also know that I'm the product of a Jesuit education. Coincidentally, I attended the same college, the College of the Holy Cross, as Tony Fauci, exactly 20 years later. The motto of a Jesuit education is men and women for others. And that's what we need to be as a community at the Department of Medicine. We need to be men and women for others. I've recommended to many of my colleagues on the leadership team that they read a book called Fraternity by Diane Brady. It's the story of 1968 when then theology professor, Father John Brooks at the College of the Holy Cross, following the death of Martin Luther King, decided that he needed to do something. And what it is it's a powerful story of a single person changing the direction of a university, of a college forever. He personally recruited that year 20 Black men, mostly from the inner cities of the Northeast, to come to Holy Cross. He then mentored them, guided them, not just during their 4 years, but throughout their lives. Among those 20 men were a future Supreme Court Justice, a future Solicitor General of the United States, a future Pulitzer Prize winner. It's an extraordinary tribute to the bond amongst these men, and it's an extraordinary lesson that one person indeed can make a difference, and I'd ask us to think about that. So before we launch Grand Rounds, I want to promise and commit that Kathy Garzio, and I think Kathy's with us today, and I will be following up with our faculty, our staff, our trainees in discussions and meetings and listening tours. And we're going to report back to the community, not just what we've heard, but what are we doing? What are we implementing? What's important to us? So in that spirit, what I'd like to ask Errol, if we could have people shut off their video and just for 30 seconds or so, let's pause. Pray if you like, reflect if that's what you'd like to do, but really let's think about what's gone on in the country. Let's remember the brutality and the passing and the murder of George Floyd. And let's pray for a better place. Thank you for that. Let me now turn it over to Errol, and Errol will introduce today's speakers and also the format of continuing to use the Q&A device to be able to engage as many of you as possible. I'm very grateful to our colleagues. I'm particularly grateful to our colleagues for Pathology who have come week after week to answer questions and to help provide new and up-to-date information. So thanks, Scott, and thanks, Ben. Really, you've been fabulous, and we need to make you honorary members of the Department of Medicine. I think Ben already is.
Pleasure.
All right, Errol.
Thanks, Bob, and I couldn't agree more. Bob, more than now than ever, we're lucky to have both you, Kathy, and all the leadership that we do have in our department. I can just say from a communication standpoint, diversity and health disparity has been a #1 topic since the very beginning. And so it's more important than ever now. And I just want to say it from a communication standpoint, for all of you listening, both in our department and outside, if there's anything we can and should be doing better to communicate all the importance of what's going on now and making sure you all are supported, please do let us know. That's our role here, so to communicate that. So please do let us know, whether it's Grand Rounds or outside of it. Now, pivoting over to, and again, next week, we're going to talk more about this with our health disparities Grand Rounds. So moving over to our main presentation today, this is a presentation that we've been looking forward to really since March, since the very beginning of this whole thing. And all of you all know that vaccine development is probably the #1 topic. I'm going to introduce 4 speakers, and I will say now, I could easily spend the whole hour talking about their bios. It's unbelievable. So I'm just going to be brief, but I'm going to say I'm very thankful to have all 4 of you here. We have Dr. Karla Kirkegaard, PhD, the Violetta L. Horton Professor of Genetics and former chair of the Department of Microbiology and Immunology, who's going to focus a lot on the science, the background of vaccine creation. Dr. Scott Boyd, who holds both an MD and PhD, is Associate Professor of Pathology and Endowed Faculty Scholar of Allergy and Immunology. And I was going to emphasize what actually Bob already did, how lucky we are. Those of you who've been coming to Grand Rounds already know Scott pretty well if you didn't before. And Scott was the guy behind this COVID serology. He's going to be talking about a question a lot is the neutralizing antibody test. He's going to talk about that today as well. But Scott, thank you so much for being with us every week and helping us answer so many questions in your realm. Dr. Bali Pulendran, carries a PhD, and he's also the Violetta L. Horton Professor of Microbiology and Immunology. And I'll just say I was reading a lot about your bio, and your bio pretty much says what you're an expert in the development of novel therapeutics and vaccines in the setting of pandemics. I mean, so your background is tremendous, and thank you so much for being with us today, Bali. And lastly, Kari Nadeau, who carries an MD PhD. She's the Naddisy Foundation Endowed Professor of Medicine and Pediatrics. She's director of the Sean and Parker Allergy Center for Allergy and Asthma Research. And now, recently, very, very lucky to have her in my division as the Senior Director of Clinical Research and Division of Hospital Medicine. I'm so happy Neera brought you in. She has been unbelievably supportive and mentoring many of our colleagues in helping research a whole new level. Kari, thank you for joining us today. Thank you to all of you guys for being here with us. And with that being said, I will turn it over to our first presenter. And I think our first—
share my screen here.
yeah, Karla, you're good. Yep.
Yeah, we'll get
Perfect.
there. We'll get there. Yeah, well, good morning, and what a privilege to be here talking to you guys. I want to apologize in advance. I'm a virologist, and my father was an MD. And when we would talk sometimes, he'd say, Karla, it is so creepy because it sounds like you're on the side of the viruses. And I want to apologize, but to be a virologist, it's like being a criminologist. You have to try to think about their problems and what they're doing. Okay, and this is also a love letter to vaccines. And the reason is summarized on this slide. So RNA viruses, as you know, what I'm going to cover today is spike biogenesis, how great vaccines are, and the problems of being an RNA virus. Okay, so an RNA virus has, so here's a list of RNA viruses and what we've been able to do about them in public health and in pharmaceuticals. So you see, the vaccines have been very successful in the first class in controlling enteroviruses like poliovirus, which is, God willing, near eradication. Hepatitis C virus is a class of viruses called Flaviviridae. And you can see that for yellow fever virus, there's an excellent vaccine that Bali actually has worked on quite a bit. And you can see several viruses there for which there are good vaccines, and only one for which there is a pharmaceutical treatment. As you know, spectacularly, hepatitis C virus now has a combination therapy that can eliminate the virus from the body, but that's the only one. And you ask, why is that? And I'm going to try to tell you that, but the answer really is mutation, mutation, mutation. And that's less of a problem with vaccines, and I'll tell you why. Okay, so there's COVID-19 virus. We know there's no vaccine and no treatment. Okay, so what is special about RNA viruses? Well, RNA viruses have two problems. One is their error rate, because you make RNA from RNA to make a negative strand and RNA from RNA to make a positive strand. The error rate is high, and we'll get into that. Poliovirus is one of the smaller of the RNA viruses. And what you see there is it to scale with the coronavirus and the dengue virus genomes. What you'll see is that the poliovirus genome is 7,500 nucleotides and one open reading frame. So that's another problem that positive strand RNA viruses have, is to get their proteins made, because they just have one piece of RNA. And so where do you start protein synthesis? Do you start it all over the genome so you can make multiple proteins, or do you start it once? And that's usually the strategy. For example, of poliovirus and dengue virus, what you see is this continuous box. That means that all the proteins are made at once in a context of a huge polyprotein that then has to be cut up. What's relevant to vaccines is that makes it kind of hard to know what any individual protein, the shape of it is on its own, because it starts out in a huge complex with a bunch of other proteins from which it's cut out. So it's folding, et cetera. Its shape is very dictated by its neighbors. Now, what I'm going to tell you is that spike protein doesn't have that problem, even though coronavirus employs the same strategy. So if you look at the top diagram, that's the size of the coronavirus genome. And you can see that there's something called open reading frame 1A and open reading frame 1B. And that open reading frame 1A is larger than dengue virus. So that's made as a huge polyprotein, has to be processed to make the individual proteins. So we don't really know what their shape is by themselves. ORF1B is made by what's called a frame shift. So you end up being a fusion protein between ORF1A and ORF1B, and you can see what a mess that would be. That's twice the size of dengue virus. It has to be processed. So how do all these other proteins, these other 11 open reading frames get made, one of which is spike, as you see in yellow there? It's very intricate, but the main thing I'm going to tell you about this is that we don't have to worry about spike too much once the mRNA gets made. Okay, so here you go. Here is all positive strand viruses. They come in as the kind of RNA that can get translated. They make negative strands with their polymerase. That's error-prone. Then they make a positive strand from those negative strands. That's error-prone. And that's how they replicate their genome. But how do they make all the messages that make all the proteins? This is what you're going to see here. Okay, so what happens is, so you have all these full-length genomes. They're very long. But what happens when you want to make spike protein, for example? What happens is you start on the negative strand here. You go up and copy through the spike protein. And then the polymerase switches over to here to make a little piece of RNA that helps translation happen. So, and that happens for every single one of these extra proteins that get made. So by the time you're done with that, the negative strand RNA replicates, you make a positive strand, and this message for spike, for example, looks just like every other message in the cell, looks just like a human message, RNA, and then when the ribosome, this is supposed to be a ribosome, hops on, it makes a protein just as though it were making actin in your cell. So we know what the shape of spike protein is. We know what it's like when it's made. We know what it's like when it assembles. So people say, oh, the life cycle of coronaviruses is so complicated. That's all true. But by the time you get to spike protein, it's made just like any other protein, and we know its shape. Okay, so like I said, this happens for every single one of these open reading frames. And by the time you get to these messages, they look perfectly normal. Right now, I'm going to focus on these proteins that come from the open reading frame 1B. RDRP means RNA-dependent RNA polymerase. NS14 is an exonuclease that improves the error rate so that coronaviruses are a little more faithful in their replication than others. Okay, so let me show you. Let's talk about error frequency, because people mean different things by it. And I want to clarify it because a lot of conversations happen like this. One of them is I've been told by colleagues, well, we don't have to worry about the error frequency in coronaviruses. And while it is true that their error frequency is lower, it's still high enough that we have to worry about things like drug resistance and even monoclonal antibody escape. Okay, so there's something that's biochemical called the molecular misincorporations. Here's a template RNA, and here's some complement base pairing to it. And we're going to ask, what kind of nucleotide gets incorporated here? And sometimes it should be a G, right? That pairs with C, but sometimes it's another base. And that's called the misincorporation frequency, and that's on the molecular level. Of course, then it's the cellular error frequency. That's mutations per cycle of growing in a cell. So so when RNA viruses are born, they're born into a lot of company. They're born into a lot of family that they have to, that's all within the cell. So let's say this little one is the one that started the infection. Pretty soon, through multiple events like this of incorporation, you get a family, some of which are mutant at a certain frequency. And at the end of that cycle, you have, for example, with poliovirus, 3 × 10⁻⁴ probability of mutation at any one site. So I'll give you those numbers again in a little while. There's an evolutionary mutation rate, and this is the only thing that's a rate because it's in terms of years. So that's when, for example, coronavirus is being spread through a population, when it goes from this person to that person to that person. Over the course of a year, you say, how many mutations does this red person have with respect to that blue person in their bodies? And that, for coronavirus, has been measured at about 1 in 1,000 mutations per nucleotide per year. So that means for each nucleotide in this 30,000 nucleotide base, there's a certain probability that it will be mutant. So these are very different numbers, and people need to be very specific when they talk about them. Last week, there was a publication of the structure of the polymerase of COVID-19 polymerase. And what you see is that it looks a lot like other polymerases. Here's HCV and here's poliovirus. What you need to know is that right here, I'll do it over here, is where the chemistry happens, in which you add, for example, that G to that C. And this is where the RNA comes out. Now, it's at this place that the errors get made, about 1 in 10 to the minus 4 per base, per nucleotide. But as you'll see, what was discovered, I'll show you what that looks like. Okay, so here you're incorporating the G right there. This is for dengue virus polymerase. Let's look at this. You're incorporating the G, and then here's where you try to incorporate a U or an A or a C, and it's, you know, not visible on this gel. Here's a coronavirus polymerase by itself, and you see it's actually error-prone by itself. Here it is putting in the right nucleotides, and look at this. It's going really fast, and it's putting in lots of mistakes. So why do people say it's not error-prone? That has to do with something that was discovered by Sasha Gorbalenia. When the first SARS virus was sequenced, he found an exonuclease there to fix those mistakes. And he found it just by virtue of sequence analysis that this exonuclease was stolen from a host a long time ago by coronavirus. And that it increases the fidelity of the polymerase, reduces the error rate, is the reason you can make such a long 30,000 nucleotide RNA. Now, interestingly, you've heard about remdesivir, and it's one of the only what's called a nucleotide analog that gets incorporated into the RNA. Remdesivir gets incorporated to the RNA, and unlike other nucleotide analogs, it doesn't get removed. And that's because there's a contact between both the exonuclease and the polymerase. So now there's talk now of maybe relatives of remdesivir that inhibit both activities at the same time. And that might be important in order to have antivirals against this. Okay, so what are these error frequencies in terms of a cycle in the cell? What's been measured for poliovirus is this. That means there are 2 mutations per genome in this family. So the parent really never makes anybody that looks exactly like it. In HCV, there's a frequency such that there, for every 2.5 genomes, there's a mistake. And SARS is, for every 9 genomes, there's a mistake. So yes, the error frequency is lower. Yes, it's a lot higher than, for example, our DNA, which is 1,000 times more faithfully replicated. Okay, so that's where the errors come from. And the question is, where do drug-resistant and antibody-resistant viruses come from? They're all born inside the cell, right? So let's say this is the parent, and it has all these progeny. They all have, a lot of them have mutations with respect to the parent. We'll say this pink one is drug resistant. We put in any kind of antiviral. This is a protease inhibitor for HCV. And so the math is usually done. Everybody else, all the wild type ones that are sensitive to the drug disappear, leaving the drug resistant virus, which can then amplify. Now, that's assuming, that whole scenario, which is usually the case, assumes that this pink genome can go it alone, can just helps itself and not the others in the cell, which isn't true. And it also assumes that these other genomes don't hurt it, more importantly. And what we've been working on, I'm actually involved in modeling drug antiviral resistance in drug development. And what we're trying to do is get these drug susceptible viruses to fight with this one, and that's reviewed here, but not relevant to this particular talk. Okay, so what you see is there can be direct selection for drug resistance in this case, which is why there are almost no antivirals to RNA viruses. Okay, what about antibodies? Why are antibodies so great? So first of all, usually vaccines elicit a polyclonal response. That means multiple epitopes are targeted. Those are illustrated by these different colored circles. Of course, almost every spike has those multiple epitopes on them, but I got tired of drawing little circles. So when you have a vaccine, it's targeting the virus on several different positions. Therefore, even if you have a genome that's mutant and doesn't, for example, recognize that epitope, that's fine because there are other antibodies that recognize other epitopes. So a multivalent antibody response is very robust with respect to mutation. There's another phenomenon, even if you have a monoclonal antibody response, that suppresses the selection of mutations. And this was found a long time ago by Esteban Domingo, who called it phenotypic masking. So let me tell you that. Let's say you have a monoclonal antibody response. It only recognizes one epitope, which we have in black, because it's going to interfere with binding to the receptor or neutralization. And you have the birth of an RNA genome that has a mutation in it that fails to recognize that epitope. So that's in red. And so you see you're going to make virions that have some spike proteins in them that cannot bind to the monoclonal antibody. But the virion is still covered with genomes, with spike proteins that can. So in general, such a structure is still sensitive to that monoclonal antibody, even though a drug-resistant, an antibody-resistant genome was born. I hope that's clear. So you get phenotypic masking, suppression of the mutations that give rise to even monoclonal antibody resistant mutations. So what am I going to say? I'm going to summarize again that the success of vaccines against RNA viruses has been spectacular relative to the development of antivirals. And I am really interested in listening to my colleagues in the hopes that this COVID-19 can be added to this list. And that's what I have to say. So thanks for listening. And I'll stop sharing now.
Great. Thanks so much, Karla. That was wonderful. I believe, Scott, are you up next?
Yes, I am. Yep, so let's see. There we go. Okay, great. So yeah, I'm going to talk about antibody responses to coronaviruses and assays for evaluating the vaccine response to SARS-CoV-2. And so just as a refresher, the coronaviruses that we talk about as being able to infect humans belong to a phylogeny of related viruses. SARS and SARS-CoV-2 are over here, and the beta coronaviruses in this group B. But there's also 4 other sort of community or commonly associated with common colds and upper respiratory infection coronaviruses, OC43, HKU1, which are also beta coronaviruses, and then these alpha ones, 229E and NL63. And so the antigens that coronaviruses are known to have are these spike proteins, but there's also a number of other ones, nucleocapsid protein or nucleoprotein, hemagglutinastrases, other envelope proteins in the membrane, and other membrane proteins, depending which ones. So as part of giving an overview for this talk of antibody responses to endemic coronaviruses, SARS and MERS, I wanted to review some of the literature that's some of the older stuff about the endemic viruses, and then also present the most recent preprint literature and just sort of give an overview of the topic. So the things I'm going to focus on are what's the time course of the antibody response, how long do the antibodies persist against each of these different coronaviruses, what is the evidence for immune protection mediated by antibodies, and then is there any evidence for harmful effects of antibodies such as antibody-dependent enhancement. So go to the next slide here. So it turns out there's sort of an interesting history to this. Starting in the 60s and continuing up until about 1990, there was sort of quite a bit of research that went on in the UK, where the British were infecting each other with coronaviruses and recording what happened, and sort of publishing some nice studies that were actually challenge studies where community coronaviruses were administered to volunteers, and immunological and clinical measurements were made about people. So this is one study you'll see often cited in this context, Karla et al. From 1990. And the data here are for people that were infected with 229E coronavirus. There was a small group in this paper that was initially sort of testing the antibody responses to individuals who were infected with 229E. And what you can see is that quite rapidly people make IgG and IgA antibody responses, and also made neutralizing antibodies to the virus within the first few weeks. What I wanted to show here is that this also gave some useful information about how long the antibody responses persist. So you can see that by the end of the year after that initial infection happened, the antibody levels dropped quite a bit, including those for the neutralizing antibodies. In that same paper, the authors talk about a second study that was done with a larger number of individuals, 15 people, and they challenged them with 229E coronavirus. And what they noticed is that 10 individuals became infected and 5 remained uninfected, as judged by whether or not they were shedding virus from their upper airway. And so the interesting observation was that the individuals who remained uninfected, with the open circles here, they tended to have higher antibody levels, both for IgA and IgG, and those remained pretty steady throughout the course of that year following their infection, or at least their challenge. Meanwhile, those who got infected, they tended to start off with lower antibody levels, but they got a boost of their titers in response to the infection. Like the ones in the earlier preliminary study set, the titers decreased over time, so that by a year they were still somewhat above where they began, but not much more. In this paper, they also talk about an observational study where they tracked, it turns out, Canadian servicemen who basically were getting infected just by community exposure to coronaviruses. And they noticed the same thing, that the antibody responses all came up relatively rapidly within a week or two, but then also decreased over time until, in some cases, they were back to baseline within a year or so. So I think this gives us some grounding for at least this particular kind of community coronavirus, what happens to the antibody titers. And as we'll see, I'll compare that to the other ones shortly. Another aspect is that in this initial challenge study, which I'm going to come back to, so keep it in mind, 8 of the 10 infected individuals also had definite symptoms of upper respiratory infection or a cold. Okay, so next. Let's make sure that's the right slide. Here we go. Good. So this is taken from a paper that was summarizing a lot of literature that was gathered in response to the SARS-CoV-1 and MERS outbreaks. And I'm just showing it here because this plot is for a number of different studies, the time to seroconversion for antibodies, IgG, IgM, and neutralizing antibodies for each of these 2 new viruses that appeared. And what you can see is that similar to what we saw with the 229E, the time to seroconversion from symptom onset is fairly similar. It's somewhere in the range of maybe 10 to 15 days, 12 to 15 days or so. And it shows some variability from study to study, but it's actually fairly predictable. Neutralizing antibodies seem to appear also in the same time frame. So this doesn't seem to be one of those kinds of infections where it takes a long time for the neutralizing antibodies to appear. Okay, and then how long did the antibodies for MERS and SARS-CoV-1 last in people? This is something that hasn't been studied as well as some of the community coronaviruses, but there are several studies that were done for MERS. And you can see that somewhat reminiscent of the 229E studies, there is quite a drop in the initial weeks after the person is infected. But it does seem as though there's a bit more of a plateauing of the antibody responses. And in some of these studies, there were relatively low levels elicited, relatively mild disease, but in others, the people had quite a high antibody level. The IgG responses were more prominent than the IgA. In the case of SARS-CoV-1, we see something that's fairly similar, except the decrease in antibodies seem to be a little bit slower. So in this case, this was they're plotting the proportion of individuals that are positive for antibodies at all, detectable ones, and then the reciprocal titer is on this axis. So you can see that over that first 3 years, there's a considerable drop in the antibodies, but it's not as rapid as was seen in the MERS or the 229E case. Okay, so then what about the question for other coronaviruses? How protective are the antibodies? Is there really evidence of resistance to infection once you have been exposed and made an antibody response? So that's what this shows is this plot that we saw before for the individuals who were subjected to 229E infection. Those individuals at the end of this year, they were actually challenged again. And in this study by Karla et al., what they found is that almost all the people got infected again. So in other words, the 5 prior uninfected individuals and 6 of the 9 prior infected individuals became infected again as judged by new shedding virus that they didn't have before. And the good thing, however, is that of all those individuals, only one of them had symptoms at that point. So this suggested that the antibody response from a prior exposure gives you some protection. You don't get as ill, but it doesn't protect you entirely from getting reinfected. Another aspect that they measured was that the viral shedding period was shorter. So that would also be a positive aspect if this applies to SARS-CoV-2, but there may be less transmissibility if the first antibody response is having some efficacy. Another study, again from this sort of like a British invasion of coronavirus infection of each other. So they, this individual's 22 volunteers were infected and had low titers to the coronavirus, the community ones. 4 people had relatively high titers to begin with. And the ones who had low titers tended to get infected at a higher proportion. Because of the low number of people that had high titer levels, this ends up not being significant. It's like p.06, I think. But it also suggests that there is some degree of protection from having high antibody levels. And then this final study, it was suggesting that if you infect individuals and then re-expose them, you can still get infected again. In this case, the answer was different than the Karla et al. Study. So in this group of 6 individuals that were initially challenged with community coronaviruses, none of them became reinfected. And so exactly why these 2 studies differ is not exactly easy to puzzle out. In this case, they were not all 229E, so there may be some difference in the viral strains that were used. But importantly, they tested whether re-challenging somebody with a different coronavirus from the one that they were originally exposed to would result in any protection. And they found that in contrast to the challenge with the same kind they had before, 5 out of the 8 people challenged with a heterologous virus were able to get infected. So if there is cross-reactivity, it doesn't seem to be all that protected in this context. Okay, so moving right along, what's the evidence for antibody-dependent enhancement of disease? I think the evidence for that is fairly thin at this point. From SARS-CoV-1, there was some in vitro data from cell culture experiments suggesting that FC-mediated interaction via antibodies could allow some cells to be infected that would not otherwise be susceptible. But to my knowledge, that hasn't really been shown in vivo context. People have noticed that there can sometimes be a correlation of the time course of disease worsening in patients with SARS-CoV-1 and seroconversion, but that's a correlation without a causative link necessarily. And a sort of counter evidence was given that when people received convalescent plasma for SARS-CoV-1, it didn't seem to cause worsening of illness, admittedly in a non-randomized, non-placebo controlled study, but I think that overall the evidence is not very strong for antibody-dependent enhancement in these coronaviruses. Okay, so what's the latest on the SARS-CoV-2 antibody responses that we hear about the most right now? As we've talked about in some of the other discussions in this series, the spike protein is one of the ones that's been favored for lots of the vaccine candidates that are being tried now. It's also a choice for many of the immunoassays, and the one that we're doing at Stanford is using the receptor binding domain, which seems to be highly immunogenic. This does seem to be the most popular topic for vaccine efforts in the SARS-CoV-2 activity. And so what are the assays we can use to measure antibodies to SARS-CoV-2? ELISAs or other sort of similar assays that measure just the total amount of antibody generated against particular antigens for the virus. Neutralizing assays, which are really important. And just to show graphically here, you take the person's plasma or serum, you dilute it serally, and you add the virus to that. You let them incubate a while. Then you expose that to cells that could potentially be infected by the virus. If the person's plasma prevents the infection, then you can tell that they have neutralizing antibodies. In practice, in many labs, this is done with pseudotype viruses that have the spike, for example, that have a safer virus to work with, so they don't have to use a BSL-3 biocontainment facility. And then there are several places that are developing these sort of blocking assays that would test whether the antibodies can prevent interaction between the spike and the receptor for the host cells. So just to complete the comparison to other coronaviruses, the time course of development of seroconversion to SARS-CoV-2 seems quite similar to what we've seen with SARS-CoV-1 and MERS. It's in that same range of around 12 days or something like that typically, and it doesn't differ much from IgG or IgM. The same thing with neutralizing antibodies that have been measured recently. That's exactly what we see in our Stanford patients. So this is the time course of IgG going up right around that same period of time. And the evidence for neutralizing antibodies to SARS-CoV-2 has really only recently started to show up in the preprint literature. So there was this study from Fudan, Wu et al. They studied 195 patients who generally had mild illness, and they found they could detect neutralizing antibodies using a lentiviral pseudotyped assay. This shows you that the time course of those appearing is similar to the appearance of the IgG or the IgM for the most part. There's a good correlation between the RBD binding activity, which is the x-axis, and neutralizing titers. And in this study, they had the conclusion that about 30% of their patients had low titers of neutralizing antibodies. 10 individuals had none at all, but the rest seemed to make higher levels of those antibodies. The question is, are the levels that you measure with this kind of assay, are they going to be protective for the real person in the real world getting exposed to the virus? Another study that came out just recently in the preprint servers is this one from Emory, Sutar et al. A different patient cohort. They actually studied patients that were more severely ill, a smaller number, 44, but their conclusion was a bit different. So they found that of the 44 individuals, all but 4 made antibody titers that were neutralizing that they thought were reasonably high. They also found that they correlated well with the RBD binding, which is also helpful for those who've been doing that kind of immunoassay and learning that kind of information. So their conclusion was sort of more optimistic that almost all the patients developed neutralizing antibodies. But of course, these were the sicker patients as well that we know make higher levels of antibodies in general. Finally, this study from Robiani et al. At Rockefeller, they studied again sort of a more mildly ill cohort of people, 149 individuals. They also used a pseudotyped assay with retrovirus. And they were looking at people a little later out than some of the other studies, so 39 days after the onset of symptoms. They're capturing their patients at a time where the IgM titers are already starting to decrease, which you can see in that panel. What they found is more similar to the Fudan study. Not everybody made neutralizing antibodies. In fact, 33% of these mildly ill or asymptomatic individuals had no detectable neutralizing titers. About 4/5 of the individuals had titers that were below 1 to 1,000. So in this context, which would represent probably most of the people that are going to get infected with SARS-CoV-2, this would suggest that the neutralizing antibody levels might not be very high from natural infection. And this just shows the correlation between the RBD binding and the neutralizing. So, what about animal studies? There have now been 2 non-human primate studies that appeared in the literature of preprint. Actually, these ones were actually published. One of them was giving a DNA vaccine that encodes a spike protein to rhesus macaques, a reasonable number of them, 35. It has 2 doses. What they found is that neutralizing antibodies were elicited, and they were actually one of the best correlates of protection when the non-human primates were challenged with the live virus. So that's not doing this paper justice, but it does seem as though neutralizing antibodies could be elicited by a vaccine, and they can potentially be associated with protection. This other one, Chandrasekhar, that was addressing the question of if somebody's infected with the SARS-CoV-2, are they then protected against reinfection? So they did that with macaques as well. And they found that indeed, the RNA levels that you see in the primary infection of the rhesus macaques are relatively high. The animals do manage to control the virus and bring them down to undetectable in their bronchoalveolar lavage specimens. When they were rechallenged, there was very little viral RNA, suggesting that the immune response had controlled the virus proliferation. They were basically neutralizing the virus. So this gave evidence that there was really a memory response. They could detect cellular immunity as well as neutralizing antibodies. So that's also maybe a note of some optimism that infection might give some protection. All right, so just to conclude this section then, data from the other coronaviruses suggest that antibodies to SARS-CoV-2 are certainly going to be stimulated, but may decrease maybe within a time course of 1 to a few years. Whether mildly ill or asymptomatic patients will produce neutralizing antibodies is still open to question in terms of whether they'll be at high enough levels to be effective. And all these assays have different thresholds and different sensitivities. So I think that it's going to take good clinical correlation with well-designed studies to really figure out which are the best assays for predicting who's really going to be protected or not. And it may have to do with how heavy the exposure is that somebody gets in the real world, for example. And we have a nice proof of concept from rhesus macaques that the vaccine efforts are likely to be fruitful, but that's also, one can't help but hope for the optimistic outcome there. Okay, thank you. I'll stop sharing my screen and we can move to the next speaker.
Thanks so much. Bali, as you hop on now and put your slides up, I just want to let people know we'll probably go over. What we'll do is, and I see a lot of great questions coming up. I asked all the presenters to please try to answer the questions in type function, and as we do in the past, we will email all the questions and answers out to everybody along with the recording of this presentation. So Bali will be next, Kari will end. We'll probably go a little over 9 o'clock. So if you have to leave, no problem. The recording of this will be available with all the questions and answers answered on our website as well. Scott, I'll turn it over to you.
Right, thank you. Can you hear me?
Yep.
Okay, great. Can you see my screen?
Mm-hmm.
Very good. All right, so thank you very much for the opportunity to talk in this session. I thought I might talk about warp speed vaccines for two reasons. Firstly, because there's an urgent global imperative to develop vaccines against COVID, but also we only have 15 minutes, so I better go through this in warp speed time.
And
well,
Bali, your video's off in case you wanted it on. It's up to you.
Yeah, I'm a little okay, here it is. Can you see me now? Okay.
Yeah.
Very good. All right, so what is Operation Warp Speed? Operation Warp Speed is the Trump administration's push to deliver 300 million doses of safe and effective COVID-19 vaccines by January 2020. This is an unprecedented challenge in the history of vaccinology. Typically, vaccines can take more than a decade, sometimes years to develop. And for some diseases like HIV, for which we've known what the causative agent is for the decades, we still have a vaccine. So we're facing unprecedented challenges in vaccinology. Now, so Scott has laid down the framework in terms of the immunology of vaccines. What are the key questions that need to be addressed? But I'd like to reiterate some of these. So clearly there's a urgent global imperative to design COVID-19 vaccines. Of course, the key question is, do infected people develop potent and neutralizing and durable neutralizing antibodies? Why is that a key question for vaccines? Because for the longest time, the paradigm for vaccine development has been, if someone gets infected with the pathogen and spontaneously controls that infection, what are the immune mechanisms that led to this control? And could we learn from natural infections and then mimic the kind of immunological mechanisms that are protected in the course of natural infection? And can we mimic these with vaccines? Secondly, does infection protect you from future infection in humans? That might seem like a very simple question to answer, but oddly enough, in the case of SARS-CoV-19, the answers are far from clear. In fact, we don't really have an answer to that question. How long does immunity last? This is a very, very important problem. Scott highlighted this in some of the challenge studies that were done in Britain. In fact, there seems to be waning immunity. Why is that so? Why is the immunity against COVID, not COVID, but other kinds of coronaviruses waning? And why is that so with coronaviruses, but with something like chickenpox or measles or mumps or smallpox? You can get immunity that lasts your lifetime. Why is that so? There's some exciting new biology that we need to understand to address this question. What are the correlates of protection? What do we mean by correlates of protection? Correlates of protection is simply a fancy word for describing what are the immunological measurements that associate or correlate with protection. And correlates of protection are typically used by vaccinologists to develop vaccines, because when you vaccinate someone, you know how much of a certain thing, say antibody levels are induced, and beyond a certain threshold, you're protected. So that is a correlate of protection. Now, the next question is important. What kind of immune response do you need for protection? Virtually every one of the 100+ vaccines that are being developed currently against COVID are predicated on the idea that a neutralizing antibody against the SARS spike protein is what you need for protection. And in all likelihood, this is absolutely likely to be true. But you see, the immune system has evolved multiple arms of immune protection. And so, for example, the mammalian immune system has two broad families, the T cells and the B cells. There's currently, to my knowledge, no real effort in mobilizing the other arm of the immune system, that is T cells, and we'll come to this in a second. How can a vaccine stimulate robust but also durable antibody? This is a key word. It's not okay for you to be protected just a month or 2 months after vaccination. You must have protection for decades after vaccination, exactly as is the case with smallpox vaccine or yellow fever or measles vaccines. I mentioned the next question, what roles do T-cell play? Scott has beautifully addressed the question of is there antibody-mediated enhancement? And then finally, this will dovetail into Kari's presentation on the development pipeline. 100 plus vaccines. Typically, I said that it can take up to 10 years, sometimes up to 15 years. Cost estimates vary, but I've heard amounts of $500 million to take a vaccine from a concept to test it in mice, and then take it all the way into phase 3 trials, up to $500 million, but these estimates vary. Surely we don't have that amount of money to be testing 100 vaccines here. So what is the new framework for vaccine testing? I want to quickly mention this because, again, I mean, Scott did show this figure from this paper from the Emory group by Mehul Sutha and colleagues. But the main point is that, indeed, infection does induce antibody responses in the majority of people. I'd like to make the following points. Firstly, if you look at this graph, this is days after symptoms onset. This is the magnitude of the binding ELISA, binding antibody titers, and this is a log 10 scale. And you see, these are incredibly variable, ranging from undetectable to 1 in 100,000. Why is that so? Why is there such an enormous variability? And if you were a vaccinologist and you said, I wanted to induce a certain amount of antibody to be protective, where would you draw your line? Would you say this much is okay? Anyone who launched a response above this would be protected? Or would you draw the line here? We don't know. And again, this is the neutralizing antibody titers. So there's incredible variability. Generally, most people develop these between 1 to 2 weeks. And in this study, they looked as far as 30 days after symptoms onset, and you can see it. Another important point Scott raised, how long does this last? How quickly does this wane? Decay rates are unknown and correlates of protection unknown. So does infection protect you from future infection? The short answer is we do not know the answer to this question, humans. However, there are a couple of non-human primate studies that were done. This one is in Press and Science. Actually, there's an online version of this in which Dan Baruch and colleagues at Harvard infected rhesus macaques. And after a primary challenge, they measured the virus in the bowel or in the nasal swab, and you can see the viral kinetics there. They rested the monkeys for about 4 weeks, and then they re-challenged with the same virus. And now you can see a greatly attenuated, negligible viral titers in the bowel, attenuated viral titers in the nasal swab. So these are encouraging signs that indeed infection can yield some sort of protection against a future challenge. Similar results in another study that's online. Well, it's not been published to my knowledge. It's actually on one of these MEDX archive, I think. But it's a very, very small study. This is with 4 monkeys, of which 2 were re-challenged. So still generally arguing for that initial challenge can give rise to protection against a future challenge. I have to say that as encouraging as these are, we have to remember the caveats here. First, re-challenge happened only 4 to 5 weeks after the primary challenge. That's a very short time. I mean, you know, ideally you want to say that challenge gives you protection for years after the initial challenge. We don't know if that's the case. Also, small numbers of animals, 4 monkeys here, and I believe there were 9 monkeys, or maybe perhaps 11 monkeys here. And then it's not clear how long immunity lasts. There should be lasts, not costs. Okay, so that's with challenge. What about with vaccines? Again, Scott mentioned this, but I want to briefly mention 3 studies in monkeys, in non-human primates. The first study was published in Science by this company in China, Sinovac, in which they had this inactivated SARS-like virus-like particle. It's an inactivated virus, essentially, in which they immunized monkeys, and they showed there was significant protection. And the data looks quite good there in terms of the viral load, the pathology in the lungs. So that seems like it's a good vaccine, and that's certainly going forward for development. It's being tested in humans. Another example of this is the vaccine from the Oxford group, which is based on the chimp-ad vector. And again, this is inducing antibodies and T cells in mice. a single immunization induced antibody and T cell responses in rhesus macaques. Neutralization titers, if you look at the data, I don't want to use the word low because it's all relative, but the titers were between 1 in 5 and 1 in 40. What did you see in the previous slide of humans who had SARS-CoV-2 infection? It varied from undetectable to 1 in 1,000. So the question is this enough for protection? T cell responses were detected, but based on comparisons with other vaccines, it's about 40 spots per million. Challenge with the virus resulted in reduced viral loads in BAL, no difference in viral loads in nasal swab, histologically normal lungs, no evidence of viral pneumonia or inflammation. And then finally, again, from Dan Baruch's group at Harvard, there's a DNA-based vaccine in which the SARS protein is encoded by the DNA and is seen to be giving good protection. So is this protection durable? In all of these studies, the challenge was done 1 month after vaccination, probably because they were in warp speed, waiting to get their paper into Science. And what is the correlate? Okay. So finally, in the last 3 slides, I want to talk about the human vaccine effort, and Kari is going to dive into this in much more depth, but just to say that there are some 100+ vaccines that are being developed, tested in animal models, of which a handful have progressed into phase 1 trial and are progressing into phase 2 trials. Of the most notable example is the mRNA vaccine by Moderna that has just begun its phase 2 trials. We don't know what the immunogenicity data looks like in the phase 1 trial, although I've heard that they're detecting neutralization antibodies in some of the subjects. Another major issue is that how do you make these vaccines globally accessible? Put simply, how do you vaccinate the planet of 7 billion people? Even the largest vaccine manufacturer, such as GSK, has a manufacturing capacity that can produce anything like a few 100 million doses of the vaccine. So the idea that there will be a single COVID vaccine is perhaps not the best idea to put forward. Rather, one might envision a situation where there are multiple 5, 10 different options of COVID vaccines that collectively could comprise the vaccine arsenal against this virus. Now, I mentioned the Moderna vaccine. So this is one remarkable feature is the accelerated timeline. Between knowing the sequence of the virus and producing the first vaccine, it took just a few weeks, they say 42 days. And Kari is going to dive into this. And again, this is a redundant slide because Kari is now going to dive into this and we don't have much time, so I want to skip this. But just to say, a handful of vaccine candidates are being tested in the clinic. Finally, I want to end with a couple of concepts here. I mentioned to you that virtually every single vaccine that's being developed is predicated on the idea that getting enough neutralizing antibodies that can block the infection of the virus is what you need. And indeed, that has served as an effective paradigm in vaccinology for the last several decades. But the question is, what about the other arm of the immune system, the T cells? Here, I'd like to highlight some recent work that we've just done in collaboration with our colleagues at Emory, in which we did a non-human primate study. This was just published just last week, in which we showed that in non-human primates, if you stimulate the T cells, these so-called tissue-resident T cells, it's just a fancy word for T cells to traffic to all the portals of pathogen entry. In other words, these are all the different sites in which a virus can come into the body, such as the mucosal sites in the lung, but also in the case of HIV, the reproductive tracts. And so in this case, what we saw was that the presence of those tissue-resident T cells greatly diminishes the quantity of antibody you need for protection. In other words, the T cells here and the antibody work hand in hand and synergistically to provide much more enhanced and durable protection. And I believe that this concept needs to be tested in the context of COVID vaccines. And then finally, I mentioned these vaccine testing pipeline, animal model testing, phase 1, phase 2, phase 3 clinical testing. The current Moderna phase 3 testing has 30,000 people. That's untenable in the context of all the other vaccines. There has to be an alternative approach. And here I'd like to mention one potential approach is to identify early biomarkers or early signatures of vaccine immunity and efficacy. So this is one example of a study that we had done several years ago in which we vaccinated people with this vaccine called the yellow fever vaccine We collected blood samples at all these different time points, and then we did an extensive analysis of the changes in the genes that were being switched on or switched off by this vaccine. This was in a drop of blood. You could do analysis of the changes in the genes, and working with computational people, we were able to define a gene signature that was switched on by this vaccine, and then correlate this early gene signature that happened just 3 days or 7 days after vaccination with the much later immune antibody response. And therefore, we could predict the magnitude of the antibody response in a completely independent trial. So it seems to me that this style of approach would be very, very useful in early screening of candidate COVID vaccines in phase 1 trials, so that you could then say rapidly, this is likely to be highly immunogenic, this is not likely to be highly immunogenic. Now, this could also be used to actually assess efficacy. So you can, in this case, this is a different pathogen, malaria, in which there's a challenge model, in which you could deliberately infect people with Plasmodium falciparum. In other words, this is how it's done. Mosquitoes infected with Plasmodium falciparum, this person is exposed to this, and then this person gets infected or gets protected, and this is dependent on their vaccine response. So perhaps one day in the future, there might be a challenge model of COVID, although the ethical reasons right now, I feel, simply do not allow that. And so finally, this really is the last slide, which will dovetail into Kari, who's going to tell us about how we're going to have a strategic approach to marshal our resources to develop all these 100 vaccines, or decide which ones go forward. Clearly, it can be business as usual, where each person tests their vaccine in a corner. There needs to be some sort of infrastructure, some umbrella, some high-level organization where all the different vaccines are tested side by side with the same standardized assays. And so this was an article written by Larry Corey and Tony Fauci and Francis Collins and John Mascola in Science that speaks to this issue. And Kari is going to tell you much more about that. So thank you.
Bali, thank you very much for that. That was a lot of great information.
Yeah.
And once you unshare, yeah, great. Kari can start sharing her slides. And again, everybody, please keep the questions coming. We will ask our presenters to answer the questions live now, and we'll also send a lot of the questions that won't be able to answer now out to you guys after an email, and then we'll get those questions answered and send it out to everybody as soon as possible with the video. We'll try to get that turnaround as quickly as possible today. Thanks for those of you who can stick with us, and we'll keep going over to Kari. Oh, Kari, I think you're muted.
Thank you, everyone. I want to appreciate your time. I'll try to go fast. Most importantly, I'm happy to share these slides. And so quickly, timeline-wise, again, the fastest vaccine's ever been done is in 5 years with Ebola, but importantly is thanks to the Emergency Use Authorization Act by the FDA, we anticipate about 15 months, and that's exciting today. I'll talk about some of those optimisms of that particular timeline, but the vaccine challenges are increasing. I wanted to just quickly highlight them. There's a number of issues with stability administration. The ideal candidate would be thermostable, capable of self-administration, patch, intranasal, oral. As Bali spoke about, as this needs to go global, we need to ensure that the current candidates are thermostable, and that's important for being able to provide the globe the necessary vaccines. Importantly is the quantity, as we spoke about today. We need to look at the number of doses, duration of protection. We need to look at multiple manufacturers and multiple successful vaccine approvals. And then distribution is also a challenge. There's going to be limited initial quantities, who should get vaccinated, when and why and how. This is also going to lead to ethical considerations. If those of you who are interested, there's a great article that was just published in the Washington Post this morning. And Steve Yaffe, my colleague, who's an ethicist at UPenn, also spoke about this particular issue, especially for the underserved. We need to ensure that there's access. There are also challenges in terms of high-risk individuals and then distribution by age. And then age in terms of vaccine responses is also critical so that we understand that better. Mark Davis and others here are working on age as it pertains to vaccines. In terms of vaccine types, there's these 5 categories that are being moved forward in clinical development right now. There's DNA-based vaccines, So the DNA is injected and you develop an immune response to that. There's an RNA-based vaccine, which is what Moderna is inclusive of, and that's important because you need the viral proteins to be translated in order for that vaccine to work. There's a non-replicating viral vector that's well established as an inactive or killed viral vector, and then you can express proteins of the SARS virus, as Karla was speaking about, to be able to elicit an immune response. And then there's the characteristic viral vaccines that we've all obtained when we were children, as well as adults. There's the inactivated viral vaccine, as well as live attenuated vaccines. This is just a pictorial graph of these different types of scenarios as to how we can best create a vaccine for COVID. So what are the candidates to date? There's over 78. Phil Pang had given a presentation recently at our grand rounds on a monoclonal antibody development. I won't be speaking about that today, but importantly, there are 8 most advanced in clinical trials. That's what I'll speak about today. So when you look at this graphic by Nature Reviews that was published in May, again, just about 2 weeks ago, you can see from those that are in the exploratory phase all the way to preclinical and phase 1, it really drops down. And to be able to get into the clinic, there's an inactivated viral vaccine. There's some positive safety data from a company called Sinovac. There are 3 trials going on in the non-replicating viral vector category in Beijing, as well as another being done at University of Oxford, which many of you probably saw in the media. That's a non-replicating viral vector as well. And then there's the RNA encapsulated vaccine by Moderna. Again, only 8 volunteers have been done so far, so tough to be able to say whether or not this is truly effective or not. And then there are other RNA vaccines moving forward by Pfizer, but no data is available yet. So what's the most possible exciting? And again, this opinion should be predicated on the fact that these are very preliminary studies. There are not many patients that are in these studies. We have to be extremely cautious with interpreting this data, and we need to make sure that we apply rigorous standards with the FDA to be able to move forward and to engage public trust in the vaccine development. So Moderna's RNA vaccine is entering phase 2 trials right now. They have shown that in 8 participants with the doses that they gave, they did show neutralizing antibody titers, and as Scott mentioned, we need to make sure that these are actually effective or not. They're going to proceed with a phase 2 to enroll 600 participants, and it looks like May 29th we'll be able to gather some more data after that date, and we're now in June, so hopefully we'll get some more data on their phase 2. Their final protocol will hopefully be starting in July. Then there are the non-replicating viral vector 3 trials, CanSino Biologics in Beijing. They are assaying their antibody production by ELISAs, like Scott had mentioned, and they have some preliminary good efficacy data. So I just want to do a shout out because of the mRNA vaccines that a lot of people are excited about in the media right now. There needs to be stability, and as we spoke about, that's really important to be able to get out and distribute. And from making a stable mRNA vaccine oftentimes means you have to pack it better into double-stranded RNA. And Nova just did a great presentation, if all of you have time to watch it, on COVID vaccines as well as COVID therapies. And Riju Das is profiled there. He's an engineer and a chemist here at Stanford, and he is creating a way to solve puzzles to be able to create RNA stability. So again, a shout out to the multidisciplinary approach that Stanford has created through our teams of research in COVID. And then finally, and this is really hot off the press, just last week, Merck, Novartis, and J&J decided to come in the game. Obviously, this is really important for large pharmaceuticals to be prepared for this warp speed program to be able to mass produce vaccines to be able to get out to the globe. And there are a number of clinical trials that they are now supporting. Finally, there is a variety of published opinion as to when a vaccine will be ready, and I want to stress published opinion rather than published data. Everything is being extrapolated on. They vary between 3 months to 18 months. Tony Fauci is cautious in saying 18 months, but there are a lot of other people that I think we need to be very careful about when we hear how long it will take, or when we talk to patients. We need to be sober and humble in the fact that this could take much longer, and we need to make sure there are safe vaccines. And in that, as Bali had mentioned, there is this wonderful accelerator that's going on right now through multidisciplinary approaches at the NIH level with the FDA and companies. And I want to stress that in the history of vaccine development that you've heard from my colleagues today, the best and the safest is not necessarily the first vaccine that comes forward in clinical development. And we need to make sure that we apply some of the principles and importantly, the rigor of clinical trials. And most likely my colleague who also consults with the FDA, Paul Offit, has mentioned that the phase 3 trial will most likely start in July, thanks to BARDA and thanks to the active NIH accelerator group. And there'll probably be 20,000 people in the active arm, 10,000 people in placebo. And he is quoted saying that it will likely end in the fall and we could potentially have a vaccine then. So again, these are exciting, optimistic characteristics of a COVID vaccine. We've talked about the status today as well as the science, and importantly is to be able to have effective assays and effective ways to monitor how well patients are doing. Finally, I want to thank you and want to thank Stanford and as well as you and your attention going past this timeline, and we'll try to answer the questions via the chat group. Thank you.
Awesome, Kari. Thank you so much for that. And really, so we're at 9:10 right now, 9:11. So we'll forego doing live questions this time, but the remaining questions I'll email to you guys and ask you to answer. And thank you for answering all the other questions for the last 20, 30 minutes or so. So with that being said, thank you everybody for joining us today. We look forward to seeing you next week. Again, to our presenters, thank you so much. And you all have a good rest of the week.
Thank you, everybody.